In this lesson, you will learn how one parent can give rise to new organisms through several different biological routes. Look across the laboratory scene. The observations do not all seem to change in the same way. That contrast gives us our puzzle. How can one parent produce new organisms in different ways? Binary fission gives two offspring. Amoeba may divide in any plane, while Leishmania divides in a definite orientation. Multiple fission can produce many after repeated nuclear division. Compare the number and direction of the divisions. Fission can produce two or many offspring. Binary fission ends with two, while multiple fission can end with many. Binary fission separates one parent into two, and its orientation can differ. Multiple fission repeats nuclear division before many offspring are produced, so the division pattern distinguishes them. The sequence explains the different outcomes. Division pattern determines the fission outcome. One division gives two, while repeated nuclear division can lead to many. Use all three panels to check the pattern. What do the Amoeba and Leishmania panels show about binary fission, and how is multiple fission different? Now connect the direction and number clues. Binary fission produces two offspring. Amoeba can divide in any plane and Leishmania in a definite orientation, while multiple fission can produce many offspring after repeated nuclear division. The image shows both the direction cue and the number of offspring contrast. Next, we will compare what happens when a body breaks into fragments with what happens when a body part rebuilds missing structures. Fragmentation in Spirogyra uses body fragments that grow into new individuals. Regeneration in Planaria uses specialized cells to rebuild missing structures and is not universal in animals. Watch what each new organism starts from. Fragmentation and regeneration are not the same. One begins with body fragments growing, while the other involves rebuilding missing structures. Spirogyra fragments can continue growing as separate individuals, whereas a Planaria fragment must rebuild missing structures using specialized cells. That is why regeneration is not simply fragmentation or universal in animals. The fragment has a different job in each panel. Regeneration rebuilds missing structures. Fragmentation instead begins with body pieces that continue growing. Use the two starting fragments as your clue. How is fragmentation in Spirogyra different from regeneration in Planaria? The panels separate the two ideas clearly. Fragmentation in Spirogyra uses body fragments that grow into new individuals, while regeneration in Planaria uses specialized cells to rebuild missing structures. The caution also shows that regeneration is not routine reproduction in all animals. Next, we will see how a small outgrowth can develop into a new individual in budding. Budding in yeast and hydra begins as a small outgrowth on the parent. The bud grows and may later separate or remain attached. The shared shape gives away the process. Budding begins as a small outgrowth. The outgrowth develops while connected to the parent. A bud starts much smaller than the parent and develops while attached. It can separate later or remain connected for a time, so the same pattern fits yeast and hydra. Follow the size change from parent to bud. A bud develops while attached to the parent. Separation may happen after the bud has grown. Focus on the feature shared by yeast and hydra. What visible change identifies budding? The repeated clue is the small side growth. Budding is identified by a small outgrowth that develops on the parent and may later separate or remain attached. Both rows show that same pattern. Next, we will examine how new plants can grow from roots, stems, or leaves in vegetative propagation. Vegetative propagation forms new plants from roots, stems, or leaves, as shown by potato, bryophyllum, and sugarcane. It is not seed formation and tends to keep desired traits with less variation. Group the examples by the plant part that starts the new growth. Vegetative propagation uses plant body parts. Roots, stems, or leaves can begin the new plant. New plants arise from one parent's vegetative tissue, so they usually keep the parent's features closely and preserve desired traits. This gives less variation, although copying variation can still occur. The similarity cue explains an important consequence. Vegetative propagation usually gives less variation. The new plants are very similar to the parent, though small copying differences can still occur. Use both the plant part strip and the crossed-out symbol. What makes new plant growth vegetative propagation? The category rule comes from the starting structure. Vegetative propagation is new plant growth from vegetative parts such as roots, stems, or leaves, not from seed formation. The potato, bryophyllum, and sugarcane examples all begin from plant parts. Next, we will look at how Rhizopus forms and disperses resistant spores from Spirogyra. Rhizopus forms many spores inside Spirogyra. When released, the spores are resistant and dispersible, so they can move away from the parent structure. The enlarged case shows where the reproductive units are stored. Rhizopus spores form inside Spirogyra. The opened case then releases the spores. Spirogyra hold many spores before release. Resistant, dispersible spores can then move away from the parent and persist better during difficult conditions. The labels explain why release is useful. Resistant spores can disperse away from the parent. The sporangium holds them before they are released. Trace the path from the round case to the released dots. How do sporangia help Rhizopus reproduce by spores? The cutaway and arrows give the whole sequence. Sporangia contain many Rhizopus spores, which can be released as resistant, dispersible units. The image shows the spores inside the case and then moving away after release. Next, we will compare all the asexual reproduction modes by parent number, reproductive structure, and variation. All the taught modes use one parent, but they use different structures or events. Their offspring are generally genetically very similar, although copying variation can still occur. Read one column for the common rule and another for the differences. Asexual modes share one parent but use different roots. The structure or event changes from row to row. The final result alone does not identify the mode. The reproductive structure or event does. All these one-parent roots usually give very similar offspring, while small copying differences can still occur. The comparison gives us a reliable identification rule. Use the reproductive structure to identify the mode. Parent number tells the broad category, while structure or event tells the specific mode. Scan one row across all three criteria. What stays the same and what changes across the asexual reproduction modes? The matrix separates the shared features from the changing ones. All the taught modes use one parent and generally produce genetically very similar offspring, while the structures or events used for reproduction differ. Copying variation can still occur. The columns make both the common pattern and the differences visible. Next, you will use visible clues to identify asexual reproduction modes in unfamiliar observation cards. The 4 cards reuse familiar clues: division into 2, growing fragments, a bud, and a sporangium releasing spores. Identify each mode from the reproductive structure or event shown. Use the clue that carries out reproduction, not just the final outcome. Reproductive clues identify asexual reproduction modes. Look for division, fragments, a bud, or a sporangium. Identify the visible reproductive structure or event, then match it to the taught mode. This avoids guessing from appearance and uses the biological clue that defines the process. The classification rule is now yours to use. Match the visible event to the taught definition. That is the most reliable way to identify the mode. Choose one card and justify it from the picture. Can you identify a mode from the visible reproductive clue? Here is one complete way to use the rule. Yes. For example, card C shows a small outgrowth developing on the parent, so it represents budding. The answer comes from the visible reproductive clue rather than overall appearance. You are now ready to use reproductive structures, processes, and variation clues to explain and identify asexual reproduction modes.